Hello, everyone. Good evening to you all. How are you all doing? So, I really hope that all your preparation is going on full swing. It's less than a week left for the D Day. Yes, welcome everybody to this Power Hours. Okay, so next 2 hours we are going to make our powerful preparation for Unit 8, some part of Unit 8 and Unit 11. So we are going to see some topics like physical mapping, microbial genetics, pedigree analysis, mutations, and from Unit 11, we'll be talking about Hardy-Weinberg equilibrium, Hamilton's rule, and phylogenetic tree. And definitely, we cannot cover each and everything, but yes, we will make sure that we cover the most important things in this last hours of preparation. And definitely, we will solve questions that is going to help you in next week's exam. Yes, are you all ready? See now, guys, we are going to solve a lot of questions. Okay, so there will be a lot of numericals also that we'll be solving. As you all know, that genetics has a lot of numericals, and even we will be talking about the Hardy-Weinberg equilibrium, which has numericals, which is full of numericals, right? So I hope all of you are ready with your pen and paper to solve the questions with me, or at least look for the solution. Try to do it. If you cannot do, definitely I'm there for help. Yes. So if you don't have your pen and paper, go right away, get it right now so that we can start. Okay. Nobody will sit without pen and paper. We need that for our session today. Yeah. Good evening, Anusha, Preeti, Krishna, Lucky, Soumya, Lakshmi, Mayuri, Arzu, Soumya. Great. So I see some Biotechnica regular students also. Good guys that you have come here. Definitely that will help you in your revision. And I hope all of your revisions are going well. Okay, excellent, Hemlata. That is what I expect from everyone. Ready with pen and paper. Okay, so let's start without wasting any time. We just have 120 minutes and we have to make the most use of it, right? Okay. And I also hope that you people have your water bottle or some snacks with you so that you don't feel hungry in between and you don't have to leave the session in between. Okay, be ready with everything. Right. So, how am I going to approach this session? Will be like we'll be doing the active revision. Okay. So we'll be taking up question, and for sure, all these questions are previous year CSI questions. So we'll be taking up questions, and then we will discuss the concept behind it. Okay. And I have taken only those topics from where multiple times question has come. Okay. In fact, you will see that the same pattern question also have come multiple times. So definitely, those are the hotspots, right? Those are the most important parts, and we have to revise that. Okay. So the first topic that we have here. So I believe in Tanushri ma'am's session you have already done with the genetic mapping. So I'll continue that, and we will start with the physical mapping. Okay. Now one technique that we do in physical mapping is the this deletion mapping. Okay. And from deletion mapping, this kind of question can be asked. So many people, okay, even in the class also, when when I discuss this kind of question, students will say that, ma'am, the question itself looks so long, so complicated, we just leave it. Okay. But guys, today I'm going to give you the trick that will make sure that, like, you will wish that this kind of question comes, because this question can be solved in less than a minute. Okay. So let's see how to do that. So the question says that the location of 5 overlapping deletions have been mapped to a Drosophila chromosome as shown below. And it is clearly mentioning, like, there is no confusion. So the horizontal lines in the above figure indicate the deleted region. Okay, so if in deletion 1, this is the line, that means the chromosome is this long. From there, this much part has got deleted. Okay. Now, recessive mutations A, B, C, D, and E are known to be located within this region, but the order of the mutations on the chromosome is not known. When the flies homozygous for the recessive mutations are crossed with flies homozygous for deletion, the following results are obtained. Okay. Letter M represents mutant phenotype, plus represents wild type phenotype. On the basis of the above data, relative order of the five mutant genes on the chromosome is. Yes, Soumya, your answer is correct. Okay, yes, we have done that in the class. Good. So that means you are revising. Yes, for everybody else, let's see how to do it. Okay. Now, this question is of deletion mapping. Okay. Now, let's visit the concept in brief. Okay, so what is this deletion mapping? So deletion mapping is a technique for physical mapping. And in physical mapping, what we do is we find out the distance between the genes in terms of base pairs, and we try to find out the actual physical distance on the gene. Okay, when we talk about genetic mapping, it is just like a bunch of gene. We want to find out whether they are linked or not. That means whether like wherever in the genome they are present, they will be present together. But where in the genome, that we cannot find out by genetic mapping. Okay, for that we need physical mapping, and deletion mapping is one such technique. So in deletion mapping, what is done is we do a cross between an individual that is homozygous for the recessive mutation for the gene of interest that we want to physically map, with an individual who is heterozygous for the deletion. Okay, now the person, so if it is heterozygous for the deletion, see what happens is, now if the gene of interest is present in the deleted region, okay, so there will be a part, like when we cross these two individuals, there will be a proportion of the progeny in the next generation that will show the mutant phenotype. Okay, why mutant? Because it has got the whole chromosome from one parent, other parent is giving the deleted chromosome. Deleted chromosome do not have any copy, so this recessive allele will show its phenotype, and that will be the recessive phenotype. Okay, now in this situation, in our question that is mentioned here, so recessive phenotype will be the M phenotype, which is mutant phenotype. Okay, whereas dominant will be the plus. All right, now if the gene of interest is not present in the deleted region, then what will be the scenario? That the entire progeny will show the dominant phenotype or the wild type phenotype here. Okay, so basically what can we conclude? That mutant phenotype produced in a cross indicates that the corresponding gene is present in the deleted segment. Okay, and if wild type phenotype is produced, means that particular gene of interest is not present in the deleted segment. Okay, I'll repeat. If a mutant phenotype is produced in the deletion mapping cross, okay, that means the particular gene of interest is present in the deleted segment. Okay, so mutant phenotype means it is present in the deleted segment. Okay, wild type phenotype, if it is produced, means it is not present in the deleted segment. Okay, now going back to our question. All right, so what we will do here is we are going to analyze this deletion map. Okay, forget about this table for the time being, just focus on the deletion map here. Okay, so in this deletion map, what we see that, so it has been segmented, right? The chromosome has been segmented into 6 segments. So if we are talking about a gene that is present in this first segment, okay, here, okay, what kind of profile it should have for all these 5 deletions? So in the first deletion, it has been deleted. So if it is deleted, okay, that means it should have the mutant phenotype. Whereas for other deletions, 2, 3, 4, 5, it is not deleted. That means for that, it should have the wild type phenotype. Do you all agree? Quickly, response. Because this is the most important part. Okay, if you understand this, then you can solve this question, as I said, in less than a minute. Okay, Soumya, Lakshmi. What about others? Can I have some more responses? Okay, Mayuri. So if a gene is present in the deleted segment, then it will be mutant. Okay, it will show the mutant phenotype. If it is not deleted, okay, if it is not present in the deleted segment, then it will show the wild type phenotype. Okay. Now, in the same way, okay, in the same way, find out what will be the expression of a gene which is present in the second segment. So it is deleted in 1 and 2. So that means in both the situation, it will be MM, whereas for rest, it will be plus, plus, plus. Okay. Similarly, for any gene which is present in the third segment will be M, M, and then plus, plus. Okay. Next one, it will be plus here, M, 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 plus. Okay. Next, plus and rest 3 will be M. The last one, first first 3 pluses and then MM. Okay. Now, once you have derived that, now go and check this table. Okay. Now you just have to match the pattern. Like 90% of your work is done. You just have to match the pattern. So what you have here, M plus 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 plus. So which gene is showing that profile? Gene B. M plus 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 plus. Okay, that means in the first position, what gene is present? B. Next one, MM plus plus plus. Which gene? C. Next one, again check the profile. MMM plus plus plus. Who is showing that? D. Okay. Next profile, plus MMM plus. There is none of the gene which is showing that. Okay. So maybe the genes that we have mapped, out of that, none of that is present in this particular segment. Moving on, plus plus MMM. So that is E. That means after D, it will be E, and finally it will be A. So the order of these genes present is B, C, D, E, A. Is it understood? Do you find this question easy now? Okay, but yes, guys who are listening to this concept for the first time, it might be a little confusing. So you need to practice. Okay, take up the same question and try to solve it again on your own. All right, that will be a good test whether you have understood it or not, whether you are able to apply it on your own or not. Excellent. Moving on. Okay. Now, this was a question from last year's CSIR exam. So this question was, it's talking about the comparison between physical map and genetic map. Okay. Now, as I was telling you that there are two different ways of mapping the genes in the genome. So it's just like if I compare it with our Google Maps. Okay. So when I say distance, okay, if I say distance between two places in the city, okay, one way of saying the distance will be in kilometers. Okay, the other way will be in terms of minutes, right? Like it is 10 minutes away, or it is 30 minutes. We often say, right, instead of using kilometers or meters, we say the distance in terms of minutes, like how much time it will take to travel from this place to that place, right? So when we like this analogy, when we compare it with physical mapping and genetic mapping, so in terms of kilometers, when we are saying, that is actually physical mapping, where we are trying to find out the distance between the genes in terms of the base pairs, which will be the actual distance between the genes. Okay, now when I'm saying in terms of minutes, okay, so there are criteria attached, whether that part, okay, this A and B point, are they in the busiest part of the city or not? Okay, so if the distance between the two places is, let's say, 2 kilometers, okay, now if that 2 kilometer is in some rural area, or in some suburban part of the city where it is not that crowded, then the same distance you can cover, let's say, in 5 minutes in your car, right? But if it is in the heart of the city where there's lot of traffic, lot of signals will be there, the same 2 kilometers can be covered in 20 minutes. Do you agree? Okay, so that is what is our genetic mapping. Okay, so even though the distance between the genes in terms of physical mapping remains the same number of base pairs, but whether the genes are highly linked, very closely linked, or they are less closely linked, or incomplete linkage is there, will depend whether the area is recombinogenic or less recombinogenic. Okay. So less recombinogenic means then at that time we will say that the distance between the two places is very less. Okay. If it is more recombinogenic, okay, then we will conclude that the distance between the two places is high. Okay, because large number of recombination is happening. But in terms of physical mapping, we might find that both of them are in the same distance, same number of base pair. Okay. So now once you have understood that comparison between the physical and genetic mapping, now let's go to our question. So the following figure represents a physical map and genetic map for 5 different genes A to E. So as you see here, on the top, it is in KB, that means it's the physical map, whereas down here, it is in CM, that is centimorgan, or like if it is map units also, then also it means it is in the genetic map units. So it is the genetic map. So which one of the following statement based on the above is correct? The region between B and C is more recombinogenic than other loci. Okay. Now, see what it is showing. B and C is recombinogenic or not recombinogenic. From where you will find? You will find it from its recombination frequency. Okay. Recombination frequency of B and C is 2. That means it is the, and compared to other regions, it is the least recombinogenic segment. So when it is saying that it is more recombinogenic, it is wrong. Do you understand, Mayuri? B and C has the least number of recombination because its recombination frequency is only 2. Recombination frequency percentage itself is the centimorgan, right? So this is wrong. Second, in comparison to the regions between A and B and regions between D and E, region between D and E is more recombinogenic. Okay, so if we compare AB and DE, okay, so AB is 10 kb, DE is also 10 kb. Okay, so the physical distance between them is same. What about the recombination frequency? Recombination frequency for AB is 9, whereas for DE is 20. Okay, so it is the same distance between them. Okay, but you are taking 5 minutes to travel from one place to another, whereas you are taking 20 minutes to travel from one place to another. Okay, so that means DE, since its recombination frequency is high, means DE is more recombinogenic. Okay, so statement B is statement number 2 is correct. Okay, let's see 3 and 4 also. So 3 says that 1 centimorgan is equal to 1 kb. Now there is absolutely no relationship between physical mapping and genetic mapping. Okay, we cannot do this kind of equation. So this is wrong. 4, if pairs are mapped between D and E, the genetic distance between D and E is likely to decrease. Okay, now that is wrong. Why wrong? Now, yesterday's class, you must have studied the 3-point test cross, right? So why did we go for 3-point test cross? Because in case of 2-point test cross, we check only the terminal genes, right? So what happens is there, if the distance between the genes is more, then there might be crossing over also occurring between them. Double crossovers also occurring between them. Now in double crossovers, if you check the terminal genes remain same as the parental. Okay. But what changes? The change will be in terms of the gene which is present in the middle. So now checking the gene which is present in the middle, we can find out that out of some parentals, there are some amount of double crossovers also. That means the proportion of parentals is decreasing. And if the proportion of parental is decreasing, means the distance between the gene is increasing. Okay. So if we map extra markers between D and E, then the distance between D and E is going to increase, not decrease. Okay, that's why all these other statements are wrong, and answer is option number 2. So you can see the explanation. So in terms of physical distance, both A, B and D are equal, that is 10 kb. However, their genetic distance is different. Genetic distance of AB is 9 centimorgan and that of D is 20 centimorgan. And in comparison, AB and DE is having more recombination. So comparison of AB, DE is more recombinogenic. Is it understood, everyone? Now, I hope with this discussion, you all got an idea about the comparison part. Okay, genetic map and physical map, how to compare them. And I took this question because like few years back also there was a question in CSIR in the same exact pattern. The numbers were a little bit different. Okay, but the same exact question has been asked. All the students, can I have some quick responses? So we are 20 minutes in our session. How are you guys feeling? Can I have some BIOs here? Are you ready for the next question? Okay, excellent. Good. Like this. I like this when you people are responding so fast. Excellent. Okay, next. Right. Now we are going into pedigree question, and this question was in last year's CSIR exam only. Thank you so much, Riva. Yes. So in this next question, it is about pedigree analysis. As you can see, the pedigree chart has been given. Again, a topic which is frequently asked in the CSIR exams. Okay, like you will hardly come to a session where not a single question has come from pedigree analysis. So in pedigree analysis, there are three kinds of questions which are all usually asked. One is about calculating the probability. One is about reading the chart and find out which all progeny will be affected, and another one is where in the pedigree it is related to pedigree, but the expression is not given in the form of pedigree chart. Instead it is given in form of words. Okay, so you have to read the words, then usually there will be a band pattern or something that is given, so you have to read the band pattern and from there you have to conclude. Okay, so we'll see this type where the pedigree chart is given, and this question was asked in CSIR last year 2022. Okay, now why did I take this question? Because it tests multiple concepts. Okay, and now it is like CSIR has always been doing that. So even though the syllabus is divided into units, but the questions are related. Okay, so this question is not related with some other unit, but in unit 8 itself multiple concepts have been combined here. Okay, so this question is saying, so let's say what is it saying. So the pedigree below is in reference to Angelman syndrome, which is caused by mutation in UBE3A gene on chromosome number 50. The gene is also paternally imprinted. The individual showing AS have not been indicated in the given pedigree. So like it's not indicated who has the disorder, who doesn't. Individual 1 does not have AS. Okay. Individuals marked with dots are carriers of UBE3A mutation. So whoever has dot in them, they are all carriers. But whether they have the syndrome or not, that is not mentioned. Okay. Which one of the following options list individual, all of whom are likely to show AS? Okay. Now in this one, like two things that you have to know. One is what is this paternally imprinted. Okay, so this paternally imprinted is coming from a phenomena called as genomic imprinting. Okay, now what is genomic imprinting? So genomic imprinting is a epigenetic process. Okay, where it is used to silence the gene through DNA methylation. So a particular segment of a gene or particular gene will be methylated, and if it is methylated, so it's like adding a tag. So if it is methylated, the gene will be silent. Okay, now this genomic imprinting happens in a parental manner. Okay, so the repressed allele is methylated while the active allele is unmethylated. Now this genomic imprinting is because of which certain genes are expressed in the parent of origin specific manner. Okay, what does that mean? That if a segment is imprinted, okay, then it depends whether you are getting that particular imprinted segment from the father or from the mother. Okay, because the imprinting pattern will be different in the parents. All right. Now, this Angelman syndrome happens because of a deletion in chromosome number 50, right? But Angelman syndrome is not the only disorder which happens because of this deletion in the region of chromosome number 15. There is another syndrome called as Prader-Willi syndrome, PWS, which also happens because of deletion in the same segment. Okay. Now, so what is the difference? Like, why? So who will get Angelman syndrome? Who will get Prader-Willi syndrome? Right? So that will depend from which parent. Okay, origin, parent of origin. So from which parent the individual is getting the particular deletion. All right. So for this syndromes, okay, Angelman and Prader-Willi syndrome, what happens? So Angelman syndrome is caused if the deletion is obtained, is inherited maternally. Okay, if the deletion is inherited from the mother, then the person will suffer from Angelman syndrome. If the deletion is inherited from father, then the person will suffer from Prader-Willi syndrome. Is this clear, everyone? So if passed from mother, what will be the consequence? Angelman syndrome. If passed from father, the consequence will be Prader-Willi syndrome. Right? Now, once you have, once you remember this concept about Angelman syndrome and Prader-Willi syndrome, you have solved the question. So what you have to check in this chart now? Who are the one who are getting the deletion from their mother? Right? So 3-1 and 3-2, they are receiving from their mother. What about 3-5? 3-5 is receiving from father. So 3-5 will not have Prader-Willi syndrome. If she has, she will, she will not have Angelman syndrome. She will have Prader-Willi syndrome. Okay, but the question is not about Prader-Willi syndrome. So skip it. And this person, 4-2. She is also getting it from her mother. So whoever is getting the deletion from their mother will have Angelman syndrome. Simple. Okay, so once you just remember that concept, again, less than a minute, right? So what will be the answer? 3-1, 3-2, and 4-2. Clear, everyone? Okay, excellent. Okay, this question we have solved it before also, but this is such an excellent example of a question that I took it up again here. So how many of you are confident in solving this question on your own? You people remember this question? If you are regular in seeing our free YouTube sessions also, then also you will be knowing this question. So can I have some responses who all are able to solve this question on their own? Okay, Mayuri. What about others? No one else? Okay, so let's see this then. So in the following pedigree, 3 STR loci A, B, and C are linked on the long arm of X chromosome in the order centromere, ABC, then telomere. Further in the table, the STR alleles representing each individual is indicated based on the above X chromosome in which of the following individuals are recombinant. The hint is also given to you that X chromosome in males will help you identify the phase of the alleles. Okay. Now there are numbers given, right? So if you see this table, there are numbers given. The first thing is we get confused about the numbers. Like the pedigree chart seems simple, but when you see these numbers, you don't know what to do, right? Yeah, Mayuri, your answer is correct. So what you have to understand here is they are not very difficult. They are not very complicated. These numbers are just the alleles. Okay. So instead of using a plus sign or a lowercase letter and uppercase letter, instead of that, numbers we have used. Okay. Now, why numbers? Because we are talking about STRs. Now, what are STRs? They are short tandem repeats. So there are particular sequences which are around 2 to 8 base pair long, and those sequences are present in all the individuals, but the number of times in a particular position in the genome, number of times those repeats are repeated will be different. Okay, so from there only we have got this number. So basically, if we see this A position, so STR in A position can have 3 alleles, if you can see. So those alleles are 4, 5, and 6. Means like it is repeated 4 times. In somewhere it might be repeated 5 times. In somewhere it might be repeated 6 times. Okay, so they are the alternate forms of that particular STR. Okay. Similarly, if you see this B position, so in B position, how many alleles are given? 7, 8, and 9. Right. So again, 3 alleles, 7, 8, and 9. For C, what is given? 1, 2, and 3. Right? So they are just the alleles of the gene. Okay. So if you consider A, B, and C instead of STR, if you find it easier to consider them as gene, so let it be. Consider them as gene, doesn't matter. But consider these numbers as the alleles of those genes. Okay. Now let's see. So who all are recombinants, you have to find out. Okay. Now for that, see, for the second generation. Okay. Now in second generation, 2 is a male. Males get their X chromosome from where? Males will always get their X chromosome from their mother, right? So who is 2's mother? 1,3. Okay, 1,3 is the mother of 2. Okay, so 681, 2 is getting from his mother. Okay, so if I have to write down the genotype, it will be 681 chromosome. Other is the Y chromosome. Okay. Now for 1, what do we have? 471 and 692, but we don't know which alleles are present in which chromosome. Okay. So now what you have to do, you have to check the genotype of the father of 21. Why? Because father has only one X chromosome, and that X chromosome without any recombination will be passed down to the daughter. Okay. So who is the father of 21? 1, 2. Okay, so in 1, 2, what all alleles are present together? 4, 9, and 2. Means for sure in this 2 1 individual, 4, 9, and 2 are present on one chromosome. Okay, now if you remove here, 4 remove, 9 remove, and 2 remove, that means the other chromosome that she is getting from her mother will have 6, 7, and 1. Do you follow, everyone? Okay, so now we have written the genes in their linked allele format also. Okay, now check for the third generation. So in the third generation, what we have? So third 3-1. Okay, so 3-1 is a female. She will get her X chromosome from father and 1 from mother. Again, from father, the X chromosome that is coming to her will come in the original form without any recombination. Okay. All right. So let me repeat. Many people have asked for a repeat. Okay. See how we wrote down this genotype. Who are they? First of all, this is the genotype of 2-1 and this is the genotype of 2-2. Okay. How did we write it down? So 2-2 is a male. Okay, so if it is a male, male will have only one X chromosome and one Y chromosome. Okay, so whatever genes are present here, 681 means 6 allele for A, 8 allele for B, and 1 allele for C are present on one X chromosome because the other one is a Y chromosome. Do you people understand that? So that's why I have written down the genotype as 681 and Y. Is this part clear? Yes? Okay. Now check for the female. Okay, so females will get one X chromosome from mother, one from father. Okay. Now again, their father also has only one X chromosome. So there is no chance of any recombination happening. The alleles will be given in the same way. Right? So what do we have here? So in this situation, I'll just clear it. Yeah. So for 2-1, okay. So 2-1's father, who is 2-1's father? 2-1's father is 1-2. Okay, so check 1-2. This is 1-2. So what alleles are present for A, B, C in 1-2? It is 4-9-2. That means this is the X chromosome. This daughter is receiving from her father. So it will come in the same format. That means one X chromosome that she has inherited from her father will have the combination A for, 4 for A, 9 for B, and 2 for C. So 4-9-2 are present in one X chromosome that 2-1 has received from her father. Okay. See guys, you just remember that these are three genes, if that helps. Okay, so just remember that they are three genes which are linked and present on the X chromosome. What is this numbers? These numbers are just the alleles. Okay, so this this A can have allele 4 or 5 or 6. This B can have allele 7, 8 or 9. This C can have allele 1, 2 or 3. Okay. So 492 is for sure coming from her father. All right. Now, if you remove 492, so the numbers are jumbled here, right? So you take out 4 because you know it is present on one chromosome with 9 and with 2. Okay. That means the other X chromosome that she is receiving from her mother will have what? 6, 7, 1, whatever is left. So that's how we have found out what is the genetic combination in the linked gene format. This is how we write the linked gene, isn't it? Where the alleles that are present on the same chromosome are grouped together. Yes or no? This is the format in which we write linked genes. Correct? Like when you write A, B, C on one and a, small b, and small c in another, what does that mean? That capital A, capital B, capital C alleles of these three genes are present together on one chromosome. Small a, small b, small c are present on a different chromosome, right? So in the same way, okay, 492 respective alleles of the respective STRs are present together on one X chromosome. The other X chromosome has 671. Clear? Everybody, can I have some thumbs ups? Yeah, Sakina, your answer is correct. Red, your answer is correct. Okay, now let's move on to the third generation. So in the third generation, what we have got, so what 3-1 is having the genotype, the numbers are given, right? So see, for A, it is only one written, one number is written, 6. Okay, so what does that mean? That only one copy of A is present? No. It means that both the X chromosome have the sixth allele for A. That means for A, at the A position, the alleles are homozygous with 6. Okay. Now, again, let's check the father, because father will give the X chromosome as it is. So, for the third generation 1, the father is, who is the father? Father is 22, this guy. Okay, so 22 has the gene, has 681. Okay, so that means this person is having 681 on one chromosome, that she is she is getting from her father as it is. So you remove 681 again, what are you left with? Another copy of 6, 9 and 2. So 6, 9 and 2, from where she is getting? For sure from her mother, right? Now check the mother. Who is mother? 2, 1. 2, 1's genotype we have written here. Does 2, 1 has any chromosome with this combination 692? No. But she can form this combination if a crossing over happens here. Then it can be 692. So when this is possible? When 3, 1 is getting a recombinant X chromosome from her mother. Okay, so 3, 1 is a recombinant. Is it understood, everyone? So in the same way, when you check the other individuals as well, okay, so I'll just clear the board a bit so that it becomes easier for us to write. Okay, for 3, 2, check for 3, 2. What about 3, 2? Now 3, 2 is a male, so he is getting 492. So he is basically has 492 on one X chromosome, other one is Y. Okay, now the 412 that he is getting from his mother. Does mother has 492 to give? Yes, she has an X chromosome with the exact same combination. Means this is a parental type, or we can say this is a non-recombinant. Okay, now for 3, 3. 3, 3 is again a boy who has only one X chromosome, that is 672. Other one is a Y chromosome. 672 is coming from the mother. Is mother having 672? No. But she can form 672 if crossing over happens here between B and C. Then this 672 combination can be there, is it not? That means this is a recombinant. And when you check 3, 4, you will find that this person is also a non-recombinant. Okay, so who all are recombinants? 3, 1 and 3, 3. Okay, so to answer this question, you just have to have two basic knowledge. Okay, one knowledge is about the recombination and how to write the link genes and how the concept of the linkage. The other thing that you have to know is the inheritance pattern of X chromosome. That X chromosome will follow the zigzag pattern of inheritance. The males will get their X chromosome only from their mother, whereas the females will get one X chromosome from father, one from mother. Okay, so with that basic information, okay, and with the info, with the concept of alleles and genes clear to you, these numbers will not bother. These numbers are just the alleles of those particular genes. Okay, everyone. So those of you who are still having confusion, I recommend that after the class, again, this lecture video will be there in YouTube. So play this and again solve it along with me. Okay, you can pause in between also at that time, and that will be helpful for your slow understanding. Okay, so that you understand the concept properly. All right, and definitely try to solve it on your own. Now, another concept which is asked from pedigree analysis is the lot score concept. Okay, so this lot score is important to know. Like nobody will ask you to calculate the lot score, but the meaning of the lot score is important. Okay, now what is this lot score? Lot stands for logarithm of odds. Okay, now this logarithm of odds, it says that it is log to the base 10 of ratio of the likelihood of the genotype or phenotypic data that we have obtained in a pedigree chart following linkage divided by likelihood of the genotype or phenotypic data assuming there is no linkage. Okay, so the ratio of that and the log to the base 10 of that particular ratio is what logarithm of odds, which we call as the lot score. Okay. Now in lot score, there's a significant value of 3. Okay. How to interpret the lot score? So lot score is represented as Z. So in lot score, now 3 is a significant value. Okay. So what does this 3 means? Okay. All right. So if we suppose that, so what is lot score? First of all, lot score is the log to the base 10 of the ratio probability that the outcome that you are getting in a pedigree chart is by linkage. Okay, and divided by the probability that the outcome is by no linkage. Okay, so if we say that the chance that the outcome is by linkage is 1,000 times more likely than the outcome to be by no linkage. Okay, then what will it become? So log base 10. So here the numerator will become 1,000. Okay, denominator will become 1. So here it is log, right? So log to the base 10. So how it will be? 10 raised to power 3. Okay, so 3 will come here. So 3 log 10 of 10. Okay, this value will be 1. So here the lot score is 3. So what does lot score of 3 means? Lot score of 3 means that the probability of getting an outcome in a pedigree chart by linkage is 1,000 times more likely than getting the outcome by no linkage. Okay, so now we go back to our question. So here it is saying that for gene pair 1, lot score is 1. For gene pair 2, lot score is 2. For gene pair 3, lot score is 3. Okay. So basically when we compare this, so what is the probability that for gene pair 1, it is linked and no link? So it is chances 10 because lot score is 1. Lot score of 10 is 1. Right, log of 10 is 1. That means the chance that you are getting the outcome by linkage is 10 times more likely than than assorting independently. Right, for gene pair 2 it will be 100. Okay, because log of 100 that is equal to 2 and 3 will mean it is 1,000 times more likely. Okay, so statement B, which says that this means it is twice more likely, is wrong. Statement C that says that the gene pair 1 and 2 can be considered linked. Now that is wrong because in lot score 3 is the significant value. So lot score of 3 or more shows enough evidence for linkage. Okay, and lot score if it is in negative, then it says independent assortment. So genes pair 3, where lot score is 3, exhibits independent assortment, is wrong. In fact, gene pair 3 exhibits linkage. Okay. So statement A and D is the answer. Okay. Right. Moving on to the next question, which is about transduction. Now, transduction and conjugation and transformation, they are used for doing the microbial gene mapping. Okay. Now, what is the concept that we use here? So for this concept, okay, so for transduction, we have to look for co-transduction frequency. Okay, so let's visit the concept first. So co-transduction frequency is like, see, suppose if two genes are transduced together, means they are passed together from the donor bacterium to the recipient bacterium via the phage. Okay, so if A and B are passed together, you see, then it is called as co-transduction. All right. So if the two genes are present close to each other, then their chance of co-transduction is higher. Okay. If the genes are present far away from each other, then the chance of co-transduction is lesser. Okay. So what can we conclude? That the, like, in terms of co-transduction frequency, the co-transduction frequency is inversely proportional to the distance between the genes. Co-transduction frequency is inversely proportional to the distance between the genes. Is it understood, everyone? It is inversely proportional, okay? This is important. So if co-transduction frequency is high, means the distance between the gene is less. If the co-transduction frequency is low, means the distance between the genes is high, right? That is what we mean by inversely proportional, is it not? Okay, if one value will be high, the other value will be less, right? Now, how do we calculate the co-transduction frequency? So, number of transductants that co-inherited both the markers from the donor divided by total number of transductants. Okay, so that is the formula for co-transduction frequency. Now, going back to this question, which was asked in CSIR 2021. So, this is the table is given to you. Okay, which of the following is the correct map of the three genes? All right, so we know that co-transduction frequency can be linked with the distance between the genes, and with that we can map the genes and we can do the mapping. All right, now see the first transduction, the first set. The donor is this, the recipient is this, just the opposite. Okay, the marker that we have selected is per F plus. Okay, now out of this two, 200 and 800, which one is the co-transductant? Can anyone tell me? Lesser will be their chance of co-transduction. Absolutely correct, Zakia. So for in this, to find out which one is your co-transduction, okay, or which one is the co-transductant, you have to look at the genotype of the donor here. Which is the donor? Donor is this, FAD-L and PERF plus. So you are already checking PERF plus means FAD-L. This is the co-transductant, not the 800, this 200. Not the plus one, Mayuri, because plus is not there in the donor. Transduction is what? Transfer of genetic material from donor bacteria to recipient bacteria via a phage. Right? Donor has only FADL, not plus, but PERF plus. So now when you are selecting PERF plus, so if you are looking for co-transduction, so which gene should have come with PERF plus? It has to be FADL because that is what is present in the donor. Okay. So the co-transduction frequency of PERF and FADL will be how much? 200 divided by the total transductance, 800 plus 200, that is 1000. So that will be equal to 0.2. Understood? Calculation part is very easy. What is the difficult part here? Difficult part is to first identify who will be the co-transductant. Is it not? And that also, if your concept here is clear, you will be able to do it very easily. Now the next one, again the donor is this. Now this time can you people tell me which class, 400 or 600, which will be the co-transductant? Quickly. Yes, guys, there are so many people in the class. I want at least response from half of the people. Absolutely correct, Soumya, Mayuri, Lakshmi, correct. Red. Yeah, Priya, correct. No, from where you are getting 200, Priya? In the next class, I'm asking. In this one, Fadel and Arrow C plus. Sandeep, Gayatri, absolutely correct. Okay, so which will be the co-transductant here? This one, because this has the Fad-L same as that of the donor. Rahul, correct. Okay, so now next what we will find out, the co-transduction frequency of Fad and Arrow. Fad-L and Arrow C. So how much it will be? 400 by 1000. Okay, so it will become 0.4. Excellent. Now in the next part, very easy. Again, same way we will find it out. So the co-transduction frequency will be arrow C and per F. Okay. So in that, now this is 500, 500. But yes, this is the number that we will be taking for co-transduction frequency. So 500 by 1000. So it is 0.5. Okay. Now see, whichever, now we have to apply that concept that co-transduction frequency is inversely proportional to the distance between the genes. So if co-transduction frequency is high, that means distance between the genes is small. If co-transduction frequency is low, means distance between the gene is high. So which two genes have the least co-transduction frequency? Per F and Fad L. Right? That means in a group of 3 genes, Per and Fad will be the genes which are farthest placed. Okay? And arrow C is present somewhere in the middle. Okay? But not exactly in the middle. So it will be present more towards which one? Fad L or arrow C? Again, by co-transduction frequency, you can find out. Like arrow C will be present. So Per and Fad, they are the farthest. So one is Per, one is Fad. Okay. Now arrow C will be present where? More towards Fad or more towards Per? So that also we can find out with their co-transduction frequency. So co-transduction frequency of this is lesser compared to this. Means arrow C will be more towards Per. How come towards Fad? Co-transduction frequency of arrow C and Fad L is 0.4. Arrow C and Per F is 0.5. Higher co-transduction frequency means lesser distance between the genes. Remember? That's right, dream narrator. So arrow C will be present more towards Per. Okay, and if you talk about the percentage of co-transduction that is given, okay, so 2 means it is 20%, 0.4 means 40%, 0.5 means 50%. So when you match it with the map given, what will be the answer? Option B. Why D will be the answer? So arrow C is, yeah, so actually, yes, so none of them are correct here if you see. Okay, so if you see the position wise, none of them are correct. But if you see by the percentage wise, okay, so arrow C and Per F, how much is the co-transduction frequency? 50%. Arrow C and Fad L, how much is the co-transduction frequency? 40%. Fadel and Paref, how much is the co-transduction frequency? 20%. Okay, so this way it is more correct. Do you understand, everyone? Here it is given as 60%. This is wrong. Okay, yeah, Riva. So this selected marker basically means is that in the recombinants, we, like in the transductance, okay, after the transduction is over, we are selecting only those cells that are per F plus. Means for sure they have received the per F plus from the donor. Okay. Now whether they have received Fadel or not, that we are selecting next. Okay. Moving on, another question from bacterial genetics. Now, in this one, we are talking about the conjugation experiment. This is a part B question, but I took it so that again you get a better understanding of conjugation here. So, in a conjugation experiment between HFR strain X and F minus cell, lac gene enters the recipient in 4 minutes, but F minus cell remain auxotropic for these different genes. The mating is then allowed to proceed for another 20 minutes and lac plus X conjugates are selected. Okay. So we are selecting only those cells which have received at least lac plus. All right. Now in all of those cells, okay, then we are checking what all other genes they have received. Okay. So in that, what we are getting, that 35% are U plus, 98% are trip plus, 10% are URA plus, 65% are glue plus, 0% are P plus, and 81% are glyc plus. Select the correct order of the genes as they enter from the choices given below. Okay, see here. So if you remember in the conjugation, like the interrupted conjugation experiment, okay, so in that, yeah, Sandeep, just give us a minute. We will finish discussing this question. I'll show you the previous slide 1 second. So in the conjugation experiment, okay, so in the conjugation experiment, what happens is that physical contact is very, very important between the donor and the recipient. If the physical contact breaks, then the transfer will stop. Okay. So how many genes are passed will depend on how long the physical contact was maintained. Okay. Now the cells, the bacterial cells that we are talking about, they are in constant motion, right? And so because of that, it becomes difficult for them to maintain the physical contact for a longer time. All right. So when the gene, the passage of the gene takes a lot of time, then in between that, the chance of the conjugation tube to break spontaneously increases. Okay. So that's why in the conjugation experiment, the interrupted mating experiment, it was found that like the number of the gene which is passed first, number of cell that is receiving that gene is higher. Okay, but for the gene which is passed last, okay, the number of cells receiving that is lesser. Why? Because in between, if the gene is passed last, means it is taking more time for its transfer, and in between that, the chance of spontaneous interruption to happen or for spontaneous breakage of conjugation tube is there. Okay, so when we apply this concept here, okay, all right, so this is the percentage of the genes which is mentioned, like which all gene is having what all percentage, right? So in that, what we have to do, so the gene which is passed first will be having the maximum number of percentage of cell receiving that gene. So here 100% are lac plus. Okay, after that, what? So we have to arrange the genes here in terms of the decreasing order of their frequency of passage, right? So trip at 98, then next number will be gly at 85, then will be glue at 65, then lu at 35, then URA at 10, and at last will be Phi at 0%. Okay, so first for sure is the Lu, because is the lac that we are selecting. Okay, so this is the order we have got. So the percentage, so this was 100%, then it was 98. Okay, this is not the minute I'm writing. Okay, this is the percentage that I'm writing. All right, so 98, then 81, then 65, 35, 10, and 0. Okay, yes, most of you have already given the correct answer. Excellent. Yes, answer is option D. Okay, now next, moving on to another concept, that is mutation. Now in mutations, yes, there are some specific areas from where questions are asked. One type of question that has asked many times, okay, like last 10 years paper also, if you pick, I will guarantee you that at least 6 times this question has been asked, and that is the mutation reversal study. Okay, now what is this mutation reversal study? So in mutation reversal study, if we do not know a chemical, like what kind of mutation it causes, so what we do, we make that we make that chemical to cause the mutation. Okay, then we use some known chemicals to do the reversal of the mutation. Okay, so if we do like that, so because we know the next step that we are doing is we are using some known chemicals, so we'll know what kind of mutation it can incorporate, and that will help us in finding out what was the basic nature of the mutation that was caused by that unknown chemical. Okay, so that is the mutation reversal study, right? So the study of reverse mutation or reversions can provide useful information of how mutagens alter the DNA structure, or what kind of mutation a particular mutagen is causing. Okay, so for this, what you have to know? You have to know the type of mutation that all the different popular mutagens can cause. Okay, you don't have to know, definitely not all the mutagens, but there are certain very popular or commonly used mutagens, and you should know what kind of mutation they can cause. Okay, so here is the list. This table have some of the list of them. So 5-bromouracil, 2-aminopurine, they are base analogs. Base analogs can cause transition mutation. Okay, so type of mutation is transition. You see, CG to TA. So C is replaced to T, G is replaced to A. Okay, so this is a transition mutation. Nitrous acid, ethyl methane sulfonate, hydroxylamine, they are also, like nitrous acid is deaminating agent, ethyl methyl sulfonate is an alkylating agent, hydroxylamine is hydroxylating agent. They also cause transition. Okay, but for hydroxylamine, it can cause only one type of transition. Okay, so it can cause only CG to TA transition, TA to CG. That transition it cannot do. Okay, so any mutation that is caused by hydroxylamine cannot be reversed by its own application. It has to be reversed by some other chemical. Okay, acrydyl orange, or you can have ethidium bromide or proflavin. They are all intercalating agents. Intercalating agents will do small or one nucleotide insertion or deletions. Okay, so if those insertion and deletions are happening in the coding segment of the gene, then it will result into frame shift mutation. Okay, now when we talk about transversions, okay, so transversions are caused by the oxidative agents. Okay, so transversion can be caused by oxidative agent, right? So now once you have this information, like what agent can cause which kind of mutation, then this mutation reversal study becomes very, very easy. So our scientists synthesized 4 new chemicals which has mutagenic potentials and named them as C1, C2, C3, and C4. He tried to analyze the nature of mutations caused by them and obtain the following result. Okay, the result is given here in the form of the table. See, so C1, whatever mutation that was caused by C1 was reversed by aminopurine. Okay, it said yes. Nitrous acid, yes. Hydroxylamine, it says some. Acrid in orange is absolutely no. Okay, so 2-aminopurine or nitrous acid, what kind of mutation they can cause? They can cause transition, even hydroxylamine also. Okay, that means C1 for sure is transition. C1 is transition. Same pattern you see for C3. Okay, C3 also can be reversed by 2-aminopurine nitrous acid. For by hydroxylamine, it cannot be. But that is possible because hydroxylamine can do only one conversion, Cg to Ta, not Ta to Cg. Okay, but still, because it can be reversed by 2-aminopurine and nitrous acid, which are producing transition, so C3 is also transition. What about C2? C2 cannot be reversed by any of them. Okay, so all these agents that we have used, so they can either cause transition or single nucleotide insertion or deletion. Okay, so C2 can neither be transition or single nucleotide insertion or deletion. Apart from that, it can be any other mutation. Okay, so it can be a transversion, it can be a large segment deletion. Okay, what about C4? C4 is getting reversed by acrid in orange. Acrid in orange can cause insertion or deletion mutation, right? That means C4 is single nucleotide insertion deletion. So answer will be option A. C1 transition, C2 transversion or large deletion, C3 transition, C4 single base insertion or deletion. Is that understood, everyone? Okay, so we have already crossed the 1 hour mark. For me, I felt like the time just flew away. What about you guys? Enjoying the session? And we have so many more concepts to revise. Excellent, Mayuri. I'm really glad that you are liking it. Now, another type of question which is asked from this mutation is the consequence of the paracentric inversion or pericentric inversion, or like it is the consequence of crossing over happening through paracentric or pericentric, like heterozygous paracentric or pericentric inverted segment. Okay, heterozygous is important because if the inversion is homozygous, that means in both the homologous chromosome has the same inversion, then there is no problem in alignment. Okay, the consequence will come because there will be a problem in the alignment if the inversion is heterozygous. Okay, so what we have here, so we will see the consequence here. I'll show you the question next. So if it is a paracentric inversion means the inverted segment do not include centromere. Okay. So again, since it is the revision segment, so I'm not going in the detail. The only thing that you have to remember is if it is the heterozygous paracentric inverted segment, the consequence will be that half of the gametes will be viable, other half will be non-viable. Why it will be non-viable? Because of deletion of some genes. Okay. Why the deletion will happen? Because it will create a dicentric chromatid and an acentric fragment. Acentric fragment will be lost, will fail to segregate during the division. Dicentric fragment will create a dicentric bridge, okay, which will break from middle abruptly, and that will result. So the gametes which will receive the chromatids which participated in the crossing over, they will be non-viable. So basically, the gametes, the recombinant gametes, they will be non-viable. Why? Because of deletion of some genes. Okay. What will be the consequence if crossing over happens through heterozygous paracentric inverted segment? Sorry, pericentric inverted segment. So it is pericentric, means the inversion includes centromere. Okay. So in this situation also, okay, the recombinant gametes will be non-viable and the non-recombinant gametes will be viable. Okay. Why the recombinant gametes will be non-viable? That will be because of deletion or duplication of genes. Okay. In this scenario, in the pericentric inversion, there is no formation of any dicentric bridge. There is no formation of any acentric chromatid. Okay. It will be both be monocentric chromatids only will be formed, but it will result into deletion of some genes and duplication of some genes. And because of that, the recombinant gametes will be inviable. All right. Now let's see the question. The question can be asked in this form. So this is taken from CSIR December 2018. So in this, you see the image is given to you. Okay. So the following, it is saying that following is the picture of an inversion heterozygous zygote undergoing a single crossing over event. The following statements are given towards explaining the consequence at the end of meiosis. Okay. So first of all, what you have to identify? You have to identify whether it is a paracentric inversion or a pericentric inversion. So quickly, can I have some responses? How do you identify whether it is paracentric or pericentric? We have to look at the loop here, okay, the cross loop that is formed. We have to look and see whether it is having the centromere or not. So which is the centromere? This dot for sure is the centromere. So is the centromere present in this loop? Yes. If the centromere is present, then what kind of inversion is this, Gayatri and Lakshmi? Pericentric. See guys, always remember IC includes centromere. Okay, pericentric inversion this is because it includes centromere. And once you have found out or once you have determined that it is a pericentric inversion, then you know the consequence. What will be the consequence of pericentric inversion? So there will be the recombinant gametes will be inviable because of deletion and duplication. Okay. So A statement says that the resultant two chromosome will have deletion and duplication, correct? B says that dicentric and acentric chromatid that won't be formed in pericentric inversion. C, the inversion does not allow crossing over to occur. So this is wrong because crossing over becomes difficult, but it can still occur through the heterozygously inverted segment. D, crossing over is considered suppressed by inversion by the acentric chromosome. Again, it is not formed here. D, all gametes formed by crossover chromatids at the end of meiosis will be non-viable. That is true. And F states that the gametes having non-crossover, the parental types, will survive. F. So answer is A, E, and F. Option 4. Excellent. Zakia, calm down. Okay. Believe in yourself. You have revised enough. All right. So if you are looking for anything new concept, then please don't do it. So even if this unit 8 is completely new for you, I will say that please do not waste your time in attending this class here and you can leave. Okay, there is no time. This is not a time to study anything new. Okay, revise. This is the time to revision. Okay, only the topics that you have studied, only the concept that you have studied, this is the time to revise them only. Okay, do not try to do anything new, and calm down, relax, and then only your efficiency will increase. Next question. An individual is having a paracentric inversion in homozygous condition. The meiotic consequences of inversion can be. Okay, so here by the mark of arrow it is mentioned, right? So this place and this place. So first of all, tell me what kind of inversion it is. It is also mentioned that this is a paracentric inversion, and we can see by the arrow mark that it is a paracentric inversion because it does not include centromere. What will be the consequence here? Now, this is an excellent CSIR type of question. Okay, so the students, the regular students of Biotechnica, we have already discussed this in the class, right? So I think all of you will remember the answer. What about other students? What do you think will be the answer here? Quickly, if I can have some responses. Yes, red, correct. Himlata, correct. Soumya, no. Okay, I'll tell you the answer. Answer will be option 4, only D. D is the only statement which is correct. What is D? All gametes will have complete genome and will survive normally. Now you will say, how can this be possible, ma'am? You just told us the consequence. But remember what I said. Those consequences will come if the inversion is in heterozygous condition. Now read the question again. It says it is in homozygous condition. So if the inversion is in homozygous condition, will there be any problem in the alignment? Not at all. Right? So if this one, so suppose if this has FED, okay, so the other chromosome also has FED, so they can straight away align. There is no requirement of formation of any loop. Right? So in this condition, yes, the gametes will have the inversion, but that will not result in the formation of any inviable gamete. Is it clear, everybody? So it is important to read the question carefully. All right, guys. So yes, there can be many more questions which can be come from Unit 8, but since we have to discuss Unit 11 also, so this is the time where we will move on to Unit 11. Okay, so Unit 11 is about evolution. It is so the most important segment, okay, where questions come is the Hardy-Weinberg equilibrium. Okay, so there can be statement based question, there can be numericals. So like every session, you can't find a single session, and this I'm not talking about just last 10 years, like last 15 years papers also. If you see, question from Hardy-Weinberg equilibrium was common, and still till date it is one such topic which has stayed common. Okay, every session we will find one or the other question from Hardy-Weinberg equilibrium, like whether in part B or part C. Now, when we look in the numericals do come in part C only. Okay, and those numericals, like they are mostly will be very, very easy. Okay, only thing is you have to understand the logic and the concept here. Okay, so let's start, everybody. Okay. Right. So let's revisit this Hardy-Weinberg equilibrium. So Hardy-Weinberg equilibrium talks about a situation where the genetic constitution, or the genetic values of a population remains constant. Okay, so what are these genetic parameters? They are allele frequencies and genotype frequency. So Hardy-Weinberg equilibrium talks about a situation where allele frequency and genotype frequency will remain constant. Okay. Now, when will it remain constant? When there is no evolutionary force acting on that population. Okay. So what are the evolutionary force? Evolutionary forces can be natural selection, mutation, migration, genetic drift, which happens by the random changes, and non-random mating. Okay, these are the conditions which has the ability to change the, or these are the evolutionary forces. Okay, these are 5 evolutionary forces which can act on the population. Okay, and when they act on the population, they have the ability to change the genetic structure of the population, right? So what are the assumptions here? Here we are saying Hardy-Weinberg equilibrium is saying that it is not going to change. So why is it not going to change? Because the disturbing influences are absent. Okay, means if we are saying that a population is in Hardy-Weinberg equilibrium, means it has the same allele and genotype frequency. So that means there is no mutation, there is no migration. Okay, there is no natural selection. The population is extremely large, because when the population becomes extremely large, the chance of genetic drift is very less or negligible, and the population is randomly mating. Okay, so these are the 5 assumptions of Hardy-Weinberg equilibrium. So the statement of Hardy-Weinberg equilibrium becomes that for a extremely large randomly mating population, the allele frequency and genotype frequency will remain constant generation after generation, provided there is no mutation, no migration, and no natural selection acting on the population. All right. Now, numerically, how this is represented. So, allele frequency we represent as P, and another allele frequency will be represented as Q. Okay. Now, it is not required that it has to be a dominant allele or a recessive allele. So, if a gene has 2 different alleles, one allele's frequency will be represented as P, other will be represented as Q. Okay. So, P plus Q, that is the addition of the individual allele frequency, will be equal to 1. Okay, so P plus Q equal to 1. Now, what about in case of genotype frequency? So, genotype frequency, it becomes P square plus 2PQ plus Q square equal to 1. Okay, now why is it so? So, suppose we have a gene, okay, gene A. Now, this A has 2 alleles, capital A and small a. Okay, now when we are saying the homozygous dominant, okay, so homozygous dominant means capital A, capital A. Homozygous recessive means small a, small a. Heterozygote means capital A, small a. Okay. So what is the frequency? What is the chance? What is the frequency of homozygous dominant? Okay. So for that, what it will be? P, allele frequency of capital A, multiplied with allele frequency of capital A. So that becomes equal to P square. Right? So the frequency of homozygous dominant is equal to P square. What about this? Frequency of homozygous recessive? Again, it will be Q multiplied with Q. So it becomes Q square. What about the frequency of heterozygote? Okay. Now, in a heterozygote, it can be like, how do we write a heterozygote? So if I write a homologous chromosome like this, so if this one has A, the other one has small a. Okay. But this is a heterozygote. Similarly, if another individual have this combination where this chromosome has small a and the other chromosome has capital A, he is equally a heterozygote, right? So there are 2 combinations in which heterozygotes can be there. One is P with Q, other one is Q with P. Okay, so that's why frequency of heterozygote become 2PQ. Do you all follow? Everyone? Okay. Now, when we compare the situation here, okay, so now when we are comparing the situation, so for genotype, so a particular gene, if it has only two alleles, it can have three different genotypes only, right? So the addition of the individual frequency will be again equal to 1. So p square plus 2pq plus q square will be equal to 1, right? Where p square is the frequency of homozygous dominant, q square is the frequency of homozygous recessive, and 2pq is the frequency of heterozygote. Clear, everybody? Now, this Hardy-Weinberg equilibrium is applicable for multiple alleles also. So, if a gene has three alleles, okay, so instead of just considering P and Q, we have to introduce one more term. Let's say R being the frequency of the third allele. Okay, so then P plus Q plus R will be become equal to 1, and for genotype, what it will be, P plus Q plus R whole square will be equal to 1. So, we have to go in the expansion of this. Okay. Now, in the same way, Hardy-Weinberg equilibrium can also be applied for polyploid condition. Okay. See here, this P plus, like this equation, if we want to simplify, what it will be, P plus Q whole square, right? So from where this square is coming? Square is coming because each individual is having two copies of each gene, right? So if the individual has polyploidy situation where more than two copies of a gene are present, right? So in that situation, this exponent will change, right? So it will become C, where C will be the ploidy level of the individual. So if you are talking about a tetraploid, for example, okay, so C becomes equal to 4. So for the genotype expansion, like in what frequency the genotype is expected in the population, it will be P plus Q. It will be the expansion of this P plus Q to the power 4 equal to 1. Okay, so Hardy-Weinberg equilibrium, as you can see, so if the conditions are true, it can be applied to any population. Okay, now let's see some numericals. Ready? So this is the point where you will be using your pen and pencil, right? Pen and paper. So this is a question from CSIR November 2020. So in a population that is in Hardy-Weinberg equilibrium, 40% of the plants are recessive homozygotes and produce white flowers. If the total number of individuals in the population is 14,000 plants, the number of homozygous dominant red flowered and heterozygous pink flowered plants will be. See, whenever this kind of question is there, so you have to remember that when a dominant phenotype is given to you, okay? So dominant phenotype can be both. It can be homozygous dominant or it can be heterozygote, okay? But when a recessive phenotype is given to you, now that is purely the homozygous recessive, right? So this is the information that you have to use. So if 40% of the plants are recessive homozygotes producing white flowers, right? That means what is Q square? Q square is 40%, right? So that is Q square is equal to 0.4. So if Q square is equal to 0.4, how much will be Q? Q will be equal to root of 0.4. Okay. So the basic computer, the standard basic computer will be provided to you in your system. So you can do this square root calculation. So it will come as, I think, 0.6 something. Yeah, 0.632. Okay, so here what you have to understand, the calculation part you can do. Okay, so here you have to understand that whenever the phenotype is given, okay, so phenotype comes from the genotype, so it is the genotypic frequency that is given to you, all right, not the allele frequency. So when it says that there are 40% white flowers and white flowers are homozygous recessives, means Q square is 40%. It is not Q. It is Q square that is 40%. Okay. So Q will be square root of 40%. Option 1. No, Mayuri, you're doing some calculation mistake. Please focus on the calculation. Answer will be option 2. Okay. So once you have calculated Q, next step will be calculating P, which is easy. 1 minus Q. Right? Okay. So now you have to calculate the number of red flowers and pink flowers. So what will be the number of, so now again, you have to calculate the number. Okay. So how do you get the number? Number you will get by frequency multiplied by the population. Population number is 14,000. Okay. So this will give you the number of red flowers. For pink flowers are heterozygote. So their frequency will be 2pq. Again multiplied with the total population. So when you substitute the values, you will get that red flower will be around equal to 1890 and the pink flowers will be 6510. Okay. Now, as you can see here that the numbers are not exactly matching. Okay. So why? But you will see that they are pretty close and there is no other option which matches this. Right? So why they are not exact match? So they are not exactly matching because there is decimal point. So anyways, we have taken till 3 decimal points. So because of that, there is the difference. Okay, but there should not be any confusion here. Because as you can see, option 1, 3, and 4, they are way off from the number that we are getting. Is it clear, everyone? Option 2, Lucky. Okay, so I am giving you the concept. You want to practice the question, you can do it after the session. Ready? Shall we move on to next question? Again, some BIOs, please. Okay. Yes. Next question. Okay. So this is again a little different, but this also has an understanding of the concept. Good to see you here, Amit. Yes. Okay. So, anyways, you can see the recording later. Let's continue. Color blindness affects approximately 1 in 12 men. Okay, 8%. So in a population that is in Hardy-Weinberg equilibrium, where 8% of the men are colorblind due to sex-linked recessive gene, what proportion of women are expected to be carriers? Okay, now see here. So in this situation, 8% is given as the individual, the men who are colorblind. Okay, now this 8% are colorblind. So I just told you that you have to look for the recessive phenotype. So color blindness is a recessive phenotype. Okay. So this 8% that is given, is it Q or Q square? So if we go by the previous question, then 8% becomes Q square, right? But this is wrong. Why? Because it would have been 8% would have been the Q square value if we were talking about any autosomal gene. Okay, but here we are talking about a sex linked or it's a X linked gene. Okay, so X linked, what happens that all the men, they will have only one X chromosome. So irrespective of what allele is present on that X chromosome, it will be expressed. So even a single copy of recessive allele, if the man is having, he will be colorblind, right? But in case of women, it is different. In women, they are having two alleles, so it will again depend whether the to produce the phenotype. It will depend whether whether the woman is heterozygote or homozygote, right? Homozygous recessive or heterozygote. So in this situation, when we are talking about the genes that are present on X chromosome, okay, the phenotype frequency of men is equal to the allele frequency of that recessive allele. I'll repeat. Okay. So in X-linked recessive disorders, males receive either the normal allele or the mutant allele in the proportion of their respective frequencies, in the proportion of their allele frequencies only in the population. So because males are hemizygous for X-linked genes, the frequency of an X-linked disorder in males is equal to the frequency of the disease allele in the population. Okay, so this is only the situation when we are talking about any gene that is present on X chromosome. Okay, so since 8% of the men are colorblind, means Q is equal to 8%. Am I clear? This would not have been the situation if we were talking about any autosomal gene. All right. So now once we have got Q is equal to 0.008, so P will be equal to 1 minus 0.08, very easy. So in case of females, the question is what proportion of females are expected to be carriers? So carriers in females will be 2pq only. All right. So now you have value of 2 multiplied with P and multiplied with Q. So this is P, sorry, and this is Q. Okay. And that will give you a value of 0.15. No, Anupam, the previous question also, if you see the calculation part is time taking, but you will be provided with the standard calculator. So with that, the calculation also will not be long. So if you just have to be careful about the numbers. So if you're doing it properly, I think in a minute is enough. 1 minute is enough to solve that question. And same for this question also. Is the concept clear, everyone? So whether it is color blindness, whether it is color blindness or we are talking about sex-linked recessive, so any color blindness or hemophilia or other X-linked traits, okay, like Duchenne muscular dystrophy. So even though it is not given in the question whether it is X-linked or not, this information you should be having. Okay, now again it is for your personal choice, Anupam. Like if you are feeling that these questions are taking more time and you can solve other questions in the same time, so definitely go for it. There is no pressure and you should not do this, that you there is no competition going on in solving the difficult questions, okay, or the lengthy questions. Definitely you have to be smart enough to do the calculation. So all part C questions are marked equally, okay. It does not matter which question you are taking, nobody is judging you. All right, so this will be your personal choice. If you feel that a particular topic is taking, a particular question is lengthy, definitely skip it. There is no point wasting time there. I'm very sorry, Himlata, we are running out of time, so I have to rush. Okay, moving on to the next question. Now, this is a question for multiple alleles. 2pq, not pq, Himlata. For women, it will be 2pq, usual, because for women, they have 2x chromosome. It is same like any autosomal genes also. Yes, so this question was also there in CSIR 2021. So sampling of 200 people for their ABO blood group was done for an urban area. The types of blood groups observed in the given population are as follows. A blood group 60 people, B blood group 32, AB 10 and O 98. Which of the following gives the correct frequency of blood group determining alleles IA, IB, and IO in the given population? Okay. Now for this, you have to have the basic information about ABO blood grouping. So A blood group, who all can be A? Who are IA, or IA, Who all can be of B blood group? IB, or IBIO. Who all can be of O blood group? Only one genotype, that is IOIO. Okay. So if I say, let's suppose that frequency of IA, let that be P, frequency of IB, let that be Q, and frequency of IO, let that be R. Okay. So O blood group means only one genotype, that is Q square. So how much is Q square given to us? 98 by 200. This is also one thing you have to remember, that this is frequency. So you have to divide it by the total individuals. Okay, so 98 by 200 is equal to Q square, okay, which will be equal to 0.49. Right? So if Q square is equal to 0.49, how much will be Q? Very easy. You don't need any calculator for that. It will be 0.7. Okay. Now you go for the elimination. So you can eliminate option 2. But yes, 1, 2 and 4, sorry, 1, 3 and 4 have Q is equal to 0.7. Okay, yes, Amit, correct. Okay, now how to proceed? Okay, now once you have got this, okay, now take only one phenotype, whether group A or group B. Okay, you can take any one of them. All right, so let's say that we are taking only blood group A. All right, so for blood group A, there are two genotypes possible. I'll show you the solution here. So if we consider A and O blood group together, okay, so then it will become what? Homozygous dominant for A, 2PR will be heterozygote for IAIO, and O blood group will be R square. Correct? Because we have assumed that frequency of IA is P, IB is Q, and IO is R. Okay. So basically, if you are considering A and O blood group, so what all genotypes you are considering? IAIO plus, sorry, write it in the order. So IAIA, IAIO, and IOIO, right? So what will be the frequency of IAIA? That is P square according to our assumption. IAIO will be 2PR, and IOIO will be R square. Okay. Now this value is not equal to 1. Remember that. Shrivani, we are dividing it by 200 because it's the frequency. There are 200 people, there are 98 people out of 200 who are showing the O blood group. So what is the frequency of the allele IO? That will be, so what will be the blood group's frequency? That will be 98 by 200. Okay, and that is Q square. So Q will be equal to square root of that. Okay, so now we have P square plus 2PQ plus R square. Now remember, this is not equal to 1 because there is one more allele given. Okay, but what will this be equal to? Okay, so what it will be equal to? It will be equal to the number of A blood groups plus the number of O blood groups divided by 200. So these two values will give you A blood group. So how much is A blood group? 60 by 200. So 0.3. How much is R square? We have already determined that it is 0.49. Okay. So whole this equation is equal to 0.79. Okay. Now when we simplify it, how it becomes? P plus R whole square. Okay. So P plus R will be equal to this. Now we already have the value of R, substitute it and we get P. and then we get Q. Okay, now definitely this is a little lengthy question, but yes, if you are preparing Hardy-Weinberg equilibrium, then this is a question that you should prepare for multiple alleles. Okay, this is an excellent example. Now moving on to Hamilton's rule. So Hamilton's rule, it talks about altruistic act where we are doing sacrifice. Okay. So whether the sacrificial act will be transmitted, will be naturally selected in the population or not, that can be determined by Hamilton's rule. So Hamilton's rule, there are three key variables: benefit to the recipient, cost to the altruist, the person who is doing the sacrifice, and the coefficient of genetic relatedness, that is R. Okay, so Hamilton's rule says that any altruistic act will be naturally selected if RB is greater than C. So what do we mean by this? That genetic relatedness multiplied with the benefit that the recipient will receive should be greater than the cost paid by the actor or cost paid by the altruist. Okay, so if we keep B and C constant, right, then what will be the situation? That higher the value of R, more is the chance of the sacrificial act, right? So more closely related to individuals are, greater is the value of altruism. Okay, now questions can be asked. So how do we calculate R? Okay, so for calculating R, so you have to understand that the relatedness between parent and offspring is 0.5. R is equal to 0.5. Similarly, between two offsprings is also, two siblings, the relatedness between two siblings is also equal to 0.5. Okay, so whether you are moving across the solid line, that is from offspring to parents, or across the dotted line, that is across the siblings, okay, the genetic relatedness is 0.5. All right, so now if the question is asked, like two different relations are given and asked what will be the value of R, or you have to find out the cost and benefit is given and you have to find out whether RB will be greater than C or not. Okay, so in those questions, in most of those questions, you have to determine R will not be given to you. Okay, so how you can determine R? So R can be determined in this way, where you uh, so number of steps that you take from moving from one person to other. Okay, so 1 by 2 raised to the power n, where n is the number of steps that you take. Okay, and steps can only be either through the solid lines or through the dashed lines. Okay, not anywhere else. Like you cannot go from here to here, that is wrong. You can either go from this to this and this to this, okay, or you can take the long way. But to find out R, you have to take the shortest way, okay, shortest path possible. So the relatedness between two individuals will be 1 by 2 to the power n, depending on how many paths you have taken. Okay, so for example, if I have to find out value of R between E and C, okay, so E to B, B to C, that means two paths I have taken, so it will be 1 by 2 to the power 2. Okay, right. Now you have a question. This is a previous year question where this is given to you already. The coefficient of relatedness between AB and AD and between DC you have to calculate. Okay, so AB means this two. So this is parent and offspring. Very easy, has to be 0.5. A and D. Okay, so to move from A to D, how you can go? You can go from D to B and then B to A, right? So how much it will be? 0.5 multiplied by 0.5. So it will be 0.25. AD will be 0.25. All right. For A to C, D to C. Okay, now how do you move across D to C? Okay, so you go from D to B, B to this, and then you go from B to C, right? So it will be equal to, so how much you are getting? 0.5 to the power 3 parts you have to take, right, for D and C. So it will be equal to 0.125. So answer will be option 1. Yes, Anupam Lakshmi, correct. Now, another format in which question can come from Hamilton's rule is where you have to modify the equation. Okay, so we know that RB should be greater than C. Okay, but that is for sure not a single way of writing the equation. You can write it in any way by moving the values on one from right hand side to left hand side or vice versa, right? So here if you see, this is in a different format it is given, okay, but the ultimate thing has to be this. All right, so you have to see which equation satisfies this format. So which of the relationship will result in an altruistic behavior? So for that it has to follow this Hamilton's equation, that is RB greater than C. So if you see this, option 3 is the only answer that will follow, right? So what has happened here, just they have moved B here. Okay, so that's why R has become equal, greater than C by B. So if you move B to this side, so again it will become RB is greater than C, which is the Hamilton's rule format that we have studied. Okay. Right, now we have last 4 minutes and I really want to do this part with you. So of the principle of maximum parsimony. Okay, now why I'm talking about this, because it is from the phylogenetic tree analysis, and this questions from this has again been asked, and in the last time in CSIR 2022 itself question has been asked, and these questions are extremely easy. Okay, so what is this principle of maximum parsimony? Okay, see, what is phylogenetic tree? We create phylogenetic tree to find out the evolutionary relationship between different species. Okay, now how do we do it? By comparing the analogous structures, homologous structures, or even with the molecular analysis we do, right? Now it is very possible that when we are trying to find out the phylogenetic relationship between 6 different species, okay, so there are two kind of relationships we get. Okay, so suppose we have got 2 different trees, so which one will be most probable? Okay, that we can find out with the help of this principle of maximum parsimony. Now, according to this principle, what it says that the simplest explanation is the simplest explanation is the most probable explanation, which has to be consistent with the facts. Okay, so what does that mean? That if 2 species, okay, so this actually considers that evolution is a very simple pathway. So if 2 species, let's say species 1 and species 2, they have any same character, okay, so there are 2 chances that they have evolved it individually, and another chance is they had an ancestor who has passed that to them. Okay, so principle of maximum parsimony says that because evolution is such a simple process, so if 2 different species are sharing the same character, then the chances are very high that they have a common ancestor who has passed down that character to them. Okay, so in case of a tree based on morphological character, the most parsimonious tree is the one that requires fewest evolutionary events to have occurred. All right, so that is what is the principle of maximum parsimony. Okay, now let me show you a question, and the question was asked in last exam itself, in 2022. So this is a cladogram. So there are 4 cladograms are given. Okay, given below, each nucleotide change is indicated by a black bar. Okay, so an evolutionary event. So what is this black bars are showing? They are showing the evolutionary event. Okay, which one of the following options represent 2 equally most parsimonious tree? So for this, what you have to see, most parsimonious tree means it is the tree having the least evolutionary event. Do you understand, guys? The most parsimonious tree will be the one having the least evolutionary events. Okay. So you see option one, how many evolutionary events it has. So basically how many horizontal lines it has, because each horizontal line is showing one evolutionary event. So this has 5, this has 4, this also has 4, and this also has 5. So which two are most parsimonious trees? B and C. I will say this takes even less than 30 seconds if you understand the concept of parsimony. Is it understood, everyone? Okay, so we are up with the time, and that's why we cannot discuss any further questions. So almost we have covered everything, whatever I was supposed to do. Maybe one or two questions I had to skip for the time constraint. So thank you everybody for joining, and I really hope that these power hours are going to help you. Tomorrow also we have another power hour for unit 2 and unit 4 taken by Divya ma'am. Please join the session, revise the concepts, and do revision constantly. Okay, this is the time for you to do time management and solve the previous year question papers. Do not try anything new right now, and keep calm, relax, take care of your health. Okay, and you will be doing good in the exam. All the best, everybody. Keep practicing, and we are here in Biotechnica, always there to help you all right, everyone. Thank you once again, everyone, for joining. And do tell all of your friends also, if they have not joined the session, please ask them to see these recordings. They will be there in YouTube. They're absolutely free, and they are excellent for your super fast revisions. Thank you, everyone, for joining. Bye bye. All the best to you all. Thank you, everyone. Good night.